Hey guys, welcome back to Bioinformatics with R. Today we're going to get into some multiple protein alignments. We're going to take different amino acid sequences, we're going to line them up and see the difference across species, and then we'll end up making a phylogenetic tree to kind of show the divergence of this gene and how it's related across species. So let's get at it. Alright, so let's start off here with loading a library that we need. MSA stands for multiple sequence alignment. Easy breezy. So let's read a string set, so the second one down, so it reads in an amino acid sequence. And I'm going to say, for me, file path get WD data sets chapter 3. This is just where I have it stored. hglobin.fa. For you, you're going to, if you're doing this on Praxis, that is, read AA strings set, and then you're just going to say hglobin.fa, because that's going to be in your my files working directory. So let's run this. So you see I have this sequence object here that has our groups or integers and all this stuff. So it's just kind of a database file. So if I type seqs and run, how about I type just seq, because that's what I saved it as. Just run that. You can see here's our amino acid sequence. So we have 8 different amino acid sequences. This is for hemoglobin subunit. And so we have a human, a mouse, a porpoise, a gorilla, a capuchin monkey, an owl parrot, a loggerhead turtle, and a pit viper. So we've got some different clades, different, a wide diversity of animals. So now let's do alignments, and we're going to call the MSA function on seqs, and our method is going to be clustal w. So there's multiple different methods here. Let's see if I can pull them up. Let's do. So this is just one of clustal w is just one of the typical protein sequence alignment tools. They have, there's actually a website that you can do load this onto as well, but we're going to do this a nice fancy way on R. Let's see, where do they have in the documentation here? It should have. Oh, I don't want to get ahead of myself. Oh. So you can see there's clustal W, clustal omega, and then muscle. And so if you can look at, if you do this in the future, you can look at what makes them different and what makes them best suitable for your alignment. We're going to go with clustal W, which is kind of the standard. So yeah. And now let's run alignments. So let's run these two. Oh, that's because I didn't run. Seqs not found. Why do I keep putting an S here? Seq. Okay, so now if we run this, you can see that it's aligned. It's the best it can. These amino acid sequence. Remember to annotate. I got to remind myself. Let's align the 8 different amino acid sequences. And I want to also point out, you can do this with either DNA or RNA as well, if you have DNA or RNA files. so any kind of alignment, it's no problem. We're doing protein just as an example here. So the next thing we're going to do is this MSA pretty print, and we're going to do that for our alignments. Our output is going to be a PDF file. We're going to say show names equals left, so to show the names of human, gorilla, etc. You can have that on or off. Show logo, so shows a sequence logo. We're going to say none. Ask for overwrite, so it's going to, every time you'd run it, it would ask to overwrite it. We're going to say false, we're just going to have it overwrite every time. Verbose equals true, so this is just going to show all the stuff as it writes it. And then we have to name the file. So the file we're going to call whole align.pdf. So to make this nicer, I'm going to, oops, put this on a page here. So every time there's a comma, I can kind of enter it down here. So I'm going to run this one. Error and ask for overwrite. Did I spell it wrong? Ask for overwrite. Oh, it's not false in quotation marks. There we go. Okay, so you see verbose, it just means it kind of shows all this stuff. Sometimes this package is a little glitchy, and I found that if I didn't call verbose, it was hanging up for some reason. So now when I open this up, you see I have a PDF in my working directory called whole align, and so as you can see here, it's an alignment of all these different sequences. So anything that has blue means it's aligned, like perfectly aligned. Anytime it's white, there's a different amino acid than the consensus, which is the majority. So pretty cool. You get to see, you know, where these amino acid mutations or changes have occurred in these different animals. And as you can see too, you would expect humans, gorillas, and capuchins, all primates, to have very closely aligned sequences that are very close to one another, which is true. And then kind of the further out, the more evolutionary distant, I should say, that you get, like an owl parrot or a loggerhead, have a lot more mutations or evolutionary divergence in their genes. So let's do MSA pretty print again, and we can do alignments. We're going to do C1030. So we're just going to take, this is the subset. I'm going to enter this down. So we're only going to take 10 through 30, those amino acids. And then we're going to say output equals PDF. And we're going to say show names equals left. Then we'll go down the next line to clear it up. So file, I'm going to say zoomed blind. Oops. PDF. Show logo equals top. And ask for overwrite equals false. And then verbose equals true. Make sure you put a comma over here. Okay, so let's run this one. Out. I don't know why I put out. Output. Okay, so now you should have zoomed align in your folder here. And I'm going to zoom in on this so you can kind of see. So as you can see, we just have the 10 through 30 amino acids. So this would be 10, this would be 30, so it's, what is that? That's 20 amino acids. And then the logo, what that means is it puts this up top here, where based on the letter size, it's I don't know necessarily necessarily the usefulness of it. It looks nice. If you got you might like the look of it. But so if it's just one big letter like this W, it means it's conserved across all 8 species. Same thing with the Y and the G and the L. If there's a few different letters, the bigger they are, the more occurrences there are. So you can see there's a big A because there's mostly A's at this 4th position, and then a small T because there's 2, and even a smaller S because there's only 1. So that's kind of how the logo thing works, and like I said, you can kind of subset it if you want. So now let's make a tree from our alignment. So we're going to have to do the library called ape and the library called sequener. And then we'll convert to a sequener alignment, which will allow us to get the distances and make a tree. So we're going to call this alignment sequener, and we'll do MSA convert the alignment to, or type is a sequener alignment. So we're calling this MSA convert package, and we're saying convert our normal alignment that we saw above and convert it into a sequener alignment. So I'm going to run, I have to run all these because I forgot to run my packages. Alignments. Okay. Now we're going to have a variable called distances. So distances1 is our sequener distance alignment. Okay, so before we get into this, let's print this real quick. So if we print our alignment, so you can see it creates a file where we have the number of comparisons, the name of each comparison, the sequence, etc., all in a what would you call it? MSA multi. What are they calling this? MSA multi something. So it's another kind of database file. And so for this, we're going to pull out the alignment and the identity from that file. So now we can say in a tree. Okay, so let's look at distances1 here. So as you can see, we have these relative distance compared to one another of all these individuals. And so this is what the tree will read. So if we do tree and we use the ape nj package, which, as you can see here, this function performs a neighbor joining tree estimation based off this publication by Saito and Nye, the original authors. And so let's do that for our distances that we just showed in this table here. And so now we have this tree, which is a list of 4. It's this is just kind of the raw data that we're going to use to draw our tree. So now let's plot our tree, and then we can call main, which allows us to plot or make the main title at the top, and we're going to say phylogenetic tree of HBA sequences. And let's run that. And zoom. So we have made a phylogenetic tree based off of our sequences here, right? So it makes sense, right? We have human and gorillas are the most identical, or at least closest related according to our 1 gene sample. A porpoise, surprisingly, is more closely related than a capuchin. This is generally why you would probably run multiple genes, right? Or entire long sequences if you were to do this to form like a much more accurate, because I would think, I'm not a primate evolutionary biologist. I know porpoise is a mammal, so it makes some sense that it's close, but I think a capuchin would be much closer. But then you have, our outgroups, as they're called, our pit viper, so a snake, a loggerhead, and a parrot are all on the outside here. So it's a good tree. I mean, I like it. It looks good, right? Interestingly enough, I heard a talk by a professor from, I think it was at College of Charleston, and there's a lot of debate of where turtles go in the evolutionary, the phylogeny, or the evolutionary tree. I don't know if this is when I was in grad school, so I'm not even going to say how many years ago. But they're kind of an interesting case. So in this case, it's weird. It's a little weird that loggerheads are closer to parrots than to pit vipers, but because pit viper obviously reptile, parrots bird. Anyways, all right, I'm rambling on. So that's it. Easy breezy. Pretty cool little set of code here if you want to make an evolutionary tree at some point. You can press your friends, right? So that's about it. I wanted to make this course a little more than just gene expression and genome stuff. This is still kind of genome stuff, but R is for everybody. There's a use. You can be a statistician, you can be an ecologist, you can be an evolutionary biologist, you can be a bioinformatician, you can be a molecular biologist, you can be whatever you want, and R has something for you. I sound like they should give me a cut, right? Like I should make some money off this. Anyways, so remember, save your file before you close. And I hope you enjoyed the video, and I'll catch you in the next one.